Those who know me know that I love signal processing. So I also love challenging students. And this is just me saying that I will challenge you a couple of times today, not in the knowledge of the deep neural networks, which are very popular, but in the knowledge of the basics in the signal processing. Okay? It will not be difficult, I promise you, but I love these challenges. And at the end, we have a short quiz for you to answer just for fun, okay? So the title of my talk today is Brain Muscle Brain Synchrony. And basically the motivation is the following, right? We know that motor unit fires asynchronously with respect to the fellow motor units. Now the pathologists can change this asynchronous behavior. We can get higher synchronization. In elicited contractions, when you apply the stimuli to the muscle, you will have a highly synchronized motor unit behavior. Not completely, but highly synchronized. In all of these cases, the motor unit discharge patterns will be sparse. It will be a point process, right? Zeros and ones. One denoting the firing, zero denoting no firing at all. And out of these basic principles, we would like to discuss synchrony between the brain and the muscle, or between the muscles in general. Okay? So this is our motivation. We saw by the talks of Jaime and Dario yesterday that the synchrony definitely exists. But we also saw that the relationship between the synchrony in the brain and in the muscle may be nonlinear, or is very likely nonlinear, and certainly complex. Okay? So this is our motivation. Motivation number two is that from the signal processing viewpoint, the whole system is really, really complex. We have multiple layers where the information can get mixed. And we have the influence of the measuring methodology. Dario presented yesterday the convolutive model of De Luca. And you saw that this convolutive model can be used for intramuscular and surface EMG recordings. Okay? So on one part, we have multiple layers combining, mixing the information from the supraspinal centers, spinal interneurons to the motor unit pools, alpha motor unit pools, and muscles, where we can measure this information. Now I will focus on the muscle interfaces in the first part of my talk. So I will not talk about measuring the nerves or the brain, right? And when we measure the muscles, we have the methodology, which has its limitations, its pros and cons, and we need to be aware of these limitations, okay? And I colored this in two different colors, in blue and in green. Why? Because my talk will be about the impact of these two different concepts. On one hand, we would like to understand the physiology, but we are limited with the eyes of the methodology, okay? And in the first part of my talk, I will focus on the methodological limitations. Now, we all know that motor units come in different sizes. They are more or less randomly distributed in the muscle tissue. Whether this random distribution is truly random or not can be discussed for various muscles. But certainly those motor units that are closer to the surface of the skin or to the uptake electrode, if you have an intramuscular electrode, produce higher energy, contribute higher energy, contribute motor unit action potentials with larger amplitudes. So the superficial will have a higher energy, and those that are close to the uptake electrodes will have higher energy. Superficial in the sense of the surface electromyogram, of course. And you can have very, very deep or very far motor units which contribute very little. And this information will become important when we will see how to measure this synchrony or synchronization between the different motor unit pools or between the cortex and a muscle. Okay? Now, regarding the measurement technology, we heard from several speakers, including Aritra on Monday, that you can measure it either on the surface of the skin or intramuscularly. And now high-density acquisitions are available in both modalities, right? This slide summarizes the basic properties of the signal that you will see if you measure it on the surface or if you measure it in the intramuscular way, okay? The frequency bandwidth is hugely different. With frequency bandwidth, it comes the length of the motor unit action potentials. And up to a few years ago, the surface methodology had the advantage of being able to observe the signal from different perspectives, from different multiple electrodes. And now the intramuscular one is catching up with the high-density intramuscular EMG, okay? But regardless of this methodology, you can compensate the MUAP shapes. You can compensate the methodology that you are using to measure the muscle, and you can go and assess the physiology. And the process of this is called the decomposition. Now, this is the second summer school of HybridNeuro. We had plenty of workshops. We had plenty of webinars. So I will skip the very details of the compensation of the measurement methodology. And I invite you to our web page, where you can see on the YouTube web page, where you can see the recordings of previous talks. So for those who are interested in the technical details, please visit our YouTube channel. There are currently 47 lectures available, and more are coming soon. And many of them you will find addressing this decomposition pipeline. In short, we have a convolutive mixing model with the motor unit pulse trains on the input and the EMG recordings at the input. There is this little fellow called noise that is playing with us, and there are the convolution kernels, which are usually called motor unit action potentials, okay? And you can reformulate this convolutive model into the matrix equation, where you gather the information about the motor unit action potential in the so-called mixing matrix. You multiply this with the vector of the motor unit pulse trains, sometimes also called spike trains. And then, of course, there is this noise, okay? And just mind the color. This is green and this is blue. This is dependent on the measuring methodology, and this is the physiology of the central nervous system, okay? Now, almost 20 years ago, more than 20 years ago, we proposed the convolution kernel compensation technique, which basically takes this equation and then inserts it into the quadratic form. So you have some pre-multiplying vector, you have the inverse of the correlation matrix of measurements, and then the EMG recordings. It combines the information of the EMG measurements in such a way that the impact of the motor unit action potentials get compensated, not fully eliminated, but compensated, and you get the quadratic form that depends solely on the pulse trains. So solely on the physiological information. Okay. Now, this first part, cross-correlation between the pulse train and the EMG signals multiplied by the inverse of the cross-correlation matrix, this part of this part of the equation is called motor unit filter. And the nice property of the motor unit filter is that when you estimate it, you can apply it in real time. It's just a matrix multiplication. So the intensive part is how to estimate this motor unit filter. And there are various techniques that has been proposed over the 2 decades that attack this challenge. Okay? Now, motor unit filters need to be estimated for each and every motor unit. And when you apply these motor unit filters to the recorded EMG signals, being intramuscular, being surface, you get this spike or pulse trains. And there is another second step, which is called the segmentation, in which you basically need to decide for each and every spike whether it represents the true motor unit discharge or basically just a noise. Okay? And it is this second segmentation step that is the most tedious and the most energy consuming, because it needs manual inspection and it needs manual editing. Okay? From the human perspective, or from the community perspective, we have challenge in front of us. On one hand, we would like to make this fully automatic because it's a tedious job. On the other hand, we would like to assure that each new generation understands the importance of this segmentation step. And to understand it, I mean to feel the pain. Okay? If you don't feel the pain, you will not pass the message to the next generation. Believe me. Okay? So do not eager for everything to be solved by the deep neural networks until you understand the problem. Okay? No pain, no gain. The motor unit filter can be made of different sizes, and we discussed across several lectures in this HybridNeuro project that the larger the size of the motor unit filter, the better the quality of the estimated spike or pulse train. Okay? In a sense, the motor unit filter is a special filter. It acts in the spatial dimensions. So this is an array of 13×5 electrodes. But it also adds the temporal dimensions. Okay? And by temporal dimension, you have a degree of freedom. You can select not to add them, not to add any history in time, or to add plenty of history in time. So you can play with the size of the motor unit filter. And as a rule of thumb, the larger the motor unit filter, the better the quality of the spike train. But large filters require also quite high processing power. And if you want to do it in real time, if you want to play with the modulations of the motor unit firing rates, like we saw in yesterday's talk, I believe, when they asked the person to modulate the firing rate of one motor unit by changing the firing rate of the other motor unit, okay? If you want to do this in real time, then it's a fair question, what is the optimal size of the motor unit filter, okay? And just to spare your time or energy, I will comment a bit on this. If you take a look at the size of the motor unit filter in terms of this mixing matrix that I introduced, you see that this temporal support here reflects in the temporal support of the motor unit action potentials in the mixing matrix. Okay? For a large temporal support, you see that all the information about the motor unit action potential shape or waveform is included. But there are these parts where the motor unit action potentials are close to zero. Are these parts adding anything to our information? Are they adding anything to the quality of the motor unit filter? That's a question. And we have another part where the shapes are close to zero. And the same thing can be done also for each and every motor unit. Okay? So can I get rid of these parts which assumingly do not contribute much to the motor unit filter? And we can make a filter shorter. This means that we cut these parts, or we can make a filter even more short. Okay? Now, probably this is too much, but there is a question: what is the optimal temporal support that you will include into the calculation of the motor unit filter? And this becomes even more important when you have fatiguing contractions where the move-up shapes change, or dynamic contractions where, due to the changes in the muscle geometry, the move-up shapes change. Okay? And of course, the quality of the motor unit filter is always judged or estimated by the quality of the pulse train that the filter can extract from the EMG. And 10 years ago, sorry, we proposed this pulse to noise ratio. We observed that, yeah, there is this jittering that should not be classified or segmented as firing, but also this jittering is valuable in the sense that it carries the information about the problems that you had. Okay? And after segmentation, you can calculate, so you have these pulses as a product of the segmentation. And then after this segmentation, you can calculate the pulse to noise ratio. And this pulse to noise ratio tells you something about the quality of the motor unit identification. And this step is essential because basically it's a fundament of the quality control for all and each application that is based on the neural codes estimated by the process of EMG decomposition. Later on, there were different derivatives of this measure, like a silhouette measure, but I prefer to call it the pulse to noise ratio because basically it's a signal to noise ratio projected into the space of the pulse trains. Okay? And with the signal to noise ratio, you immediately understand that this is not an exact measure, but basically a recommendation. Okay? And the same limitations apply to the pulse to noise ratio. Okay? Now we showed that statistically looking, it correlates very well with the sensitivity, and it inversely correlates with the false alarm rates. So it is relatively reliable measure. Statistically looking, there are exceptions, and we commented these exceptions extensively also in the first summer school last year. Again, those who didn't participate, you are most welcome to visit our webpage, our YouTube channel, and replay the lecture about this. And this pulse to noise ratio can be used to ask or to try to answer the question, what is the optimal size of the motor unit filter? And the principle is always the same, being in the highly fatiguing contractions, in the dynamic contractions, in the isometric contractions. You can use the pulse to noise ratio as a quality measure of the pulse train to assess the quality of the motor unit filter. Okay? And you have an example here for one motor unit. You see the dimension of the filter on the x-axis, and then the pulse to noise ratio of the pulse train. for each and every size of the filter. So for each and every size, we constructed the filter. We applied the filter to the EMG signals. We got the pulse train. With the very small filters, we got this yellow, ugly, not high quality pulse train. With the largest filters, we got a relatively nice pulse train, okay? And everything in between. But what you can observe is that you have these sudden jumps, right? Sudden jumps where the pulse-to-noise ratio increases quite a bit. And you can denote this sudden jump by the different size circles. So the bigger the jump, the bigger the size of the circle. And then you can plot these circles in a topological manner, where you have a spatial dimension, so columns and rows of the electrode, and the temporal dimensions. And you can then observe which channel, which delayed version of channel is the most important for this particular motor unit, for the quality of this spike train. And what you find out, if you do that, is that the results differ for each and every motor unit, okay? So you can optimize the size of the filter, but it will be motor unit specific, okay? And even though there was this section of the MUAP which was close to the zero, it was motor unit specific. For another motor unit, you would need to adapt this section differently, okay? So motor unit filter optimization, when it comes to the minimization of the computational costs, is motor unit specific. Now, this gives you still the chance to optimize in a group-wise manner. I know you could ask yourself, for these three motor units, is there a minimum set of the channels which satisfy the quality, the required quality of the motor unit filter? So you can still decide how to optimize the filter. Okay. With this, I will stop explaining the decomposition pipeline. Again, I will invite you to our YouTube channel to see all the lectures, comment on them, make it a vivid community, please. We will highly appreciate it. And now I will go to the second part, to the physiological part. So let's assume now that we relatively, in a good manner, took care about the compensation of the methodology, and then we ended up with the spike trains, and now we would like to estimate the synchronization between the different motor unit pools, between the cortex and the muscle. Okay? So we will do it in several steps, and one of the most frequently used measures, we saw it in the talks on Monday and yesterday. One of the most useful measures is coherence. Okay? Coherence tells us something about how well do the two time series match at a particular frequency. Okay? And Jaime and Dario presented yesterday that when you calculate the coherence of motor units, or between motor units and the cortex, or between the two different motor unit pools, it is wise to do it through the means of cumulative spike train. Okay? You sum together the spike trains or pulse trains, and this filters out the intrinsic jittering introduced by each and every motor unit, and leaves you with the common component the component that is common to all the motor units. Okay, we saw this yesterday, and let's do it. Let's apply it. So we have here two decomposition results from sibling muscles. You can call them sisters, you can call them brothers, but they like to dance to each other. Okay, one is gastrocnemius medialis, and the other one is gastrocnemius lateralis. Okay, and this was plantar flexion at 30% of maximum voluntary contraction. Okay, relatively high number of identified motor units, and we can calculate the coherence between the cumulative spike trains. But we can do it in different way. I can take one motor unit from gastrocnemius medialis and one motor unit from gastrocnemius lateralis, or two from each muscle, or three from each muscle, or 4, 5, up to the maximum number of muscles that I identified. Oh, sorry, motor units that I identified. Okay? And I color the coherences of these cumulative spike trains by different colors. And you can see that more motor units you take, more you articulate, more visible the peaks, the frequency peaks become. Okay? So it is always of paramount importance, as we will see, that you take relatively high number of motor units into calculation of the cumulative spike train. Now, this increase of the coherence value can be plotted also in a different manner. For each peak here, and there are 9 peaks if you count them here, you can plot the value of the coherence as a function of the number of motor units that were taken into account when calculating the cumulative spike train. And this is very simple but very useful trick. I'm sure that many of you know them, but since there are new generations, it doesn't hurt to repeat this trick. You can easily extrapolate this line and imagine what will happen, or what would happen to the coherence if you would increase the number of motor units even further. And if this line starts saturating, then you are in a position to claim that you have high enough number of motor units. If this line does not saturate, it keeps on increasing, and it seems likely that it would increase even further. Then basically you don't have enough motor units to claim the true coherence value. Okay? And look, in this case, we have 35 motor units. It's not super high, but it's relatively high. You don't get 35 motor units from each and every muscle, right? From tibialis anterior, yes. From gastrocnemius muscles, yes. Pectoralis, masseter, back muscles, forget it. Okay? This saturation, assessment of saturation, can come handy when you judge the quality of your coherence estimation. Okay? And you see that different frequency peaks demonstrate different behaviors in this plot. Okay? So this can suggest something about the robustness of coherence estimation. But the decomposition is not perfect. The segmentation is not perfect. We as humans are not perfect. We do mistakes. We do errors. And we are challenged by so-called merging of the motor units into the pulse trains. Okay? You see here a pulse train. You see with the red circles the result of segmentation. And if you pay close attention, you see that there are smaller spikes here that were not selected as discharges. There are some small spikes even here. And in this example, it is most obvious that we have more than one motor unit present in the pulse train. Now, this has been systematically evaluated by Matthias, who is sitting there and has a poster in the poster section, showing that this merging of the motor units happens due to the similarity of the motor unit action potentials, and the similarities of the motor unit action potentials derive from the similarity of motor unit properties. In fuzzy form muscles, you need to have shared location, approximately shared size of the motor unit territory, position of the innervation zone, and the conduction velocity to have a similar move-up shapes. And even though it seems unlikely that you will have these matches, it happens, and it happens frequently. Okay? And it happens at each and every contraction level. So we have a different contraction level here shown. The blue territories are those that were identified. The pink ones, those that were not identified in the process of the decomposition, and the yellow ones, those that were merged together. Okay? And what you see is that they match, at least in the location and in the size of the motor unit territory. You cannot see the effect of the conduction velocity of the innervation zone in this slide. If you increase the contraction level, the problem of the motor unit merging will increase. And this is simply the size principle, because at the higher contraction levels, you will have more motor units recruited. Okay? So, you can ask very simple question, and this is a summer school, so I'm being a bit more didactic, right? This is not a conference where I would claim what we did in the last year or so. So you can ask yourself, how sensitive is then the coherence to the decomposition errors? Because they will happen. They do happen. Okay? And you can have different types of errors. For example, you can miss motor unit firing, motor unit discharge. Okay? You can fail to segment it. What happens? What happens to each and every of these peaks in the frequency domain? So let's say if you don't have any errors, let's assume, then you have the image like this. We saw it, right? If you miss the discharges, 5% discharges in each and every motor unit. So this is quite a big error. 5% of discharges were missed in each and every motor unit, and you can see the number of motor units here. What happens? Not much. UPA. We can do the errors. Can we do even better? Can we miss 10% of motor unit discharges? 15? 20? 25? 30? 35? 40? 50? Come on, 50. 50% of the discharges missed. Not bad, right? Not bad at all. But these are not the only types of the errors that you can make. Okay? You can misplace the firing. Okay? You can incorrectly time the motor unit discharge. So let's see, maybe we are lucky again. Maybe the coherence is robust also to this type of errors. In this slide, it's 0% of motor unit discharges misplaced for plus minus 2.5 milliseconds. It was a Gaussian distribution with the standard deviation of 2.5 milliseconds. It was simulation. These are the real decomposition results, but the errors are simulated, okay? This is what we did. 2.5 milliseconds is not much, right? It's depends on the sampling frequency, but it's a very little error. If I put it to 5% of motor unit discharges, observe the change. 2.5 milliseconds in 5%. Okay? 10%, 15, 20, 25, 30, 35, 40, 45, 50. We see that in both cases that I demonstrated, you don't want to have too low number of motor units, right? Everything is below the significance level for the red lines, which have been calculated by a very low number of motor units. But if you have high number of motor units, then the coherence is robust also to these small errors. But the error of 2.5 milliseconds is really small, right? In reality, you can have much higher errors. You can put it to 5 milliseconds. 5% of motor unit discharges misplaced by plus minus 5 milliseconds. What happens? The high frequency peaks in coherence disappear completely. Okay? This one seems to be robust, but we can go on. We can increase the error. 7.5 milliseconds, 10 milliseconds, 12.5, 25 milliseconds. You get a flat line in 5% of motor unit discharges. Okay? Now this guy, this is a phantom coherence, right? Because the spectral content at the frequencies between 0 and, I don't know, 5 Hertz, it's basically filtered out, okay? It's 0 divided by 0 or something divided with very, very small. So very small value divided by very small value, okay? It's a phantom coherence. But this phantom coherence seems to be very robust to the errors. Of course, it does not depend on the discharge pattern at all, right? So you can do, I don't know, I had the result for 100 milliseconds, and apparently I didn't include it. Okay. Okay, so this gives you an insight into sensitivity of the coherences to the errors in the decomposition. Okay? We saw that if you have a high number of motor units, then you are in relatively robust waters. when it comes to the coherence estimation. And I also showed you that even with 30, 40 motor units, the coherence does not saturate, that you would profit more if you would add more motor units. Okay? But the number of identified motor units is usually limited, especially in the complex muscles, including biceps brachii. Okay? So what can we do? We can have a subject where the decomposition will yield very few motor units. We can have a muscle where the decomposition results will be poor. What can we do? We can go and analyze again why we have this small number of motor units. And the answer again is the motor unit merging. Okay? It is not that motor units are not identified. It is that they are not identified down to the level of individual motor units. What the decomposition identifies is clusters of motor units. Which clusters? Those motor units that are anatomically close to each other. Okay? This happens. And if you go and insist on the individual single motor unit analysis, then you are throwing away a lot of motor units. If you don't allow these clusters to be included in your analysis, then you are throwing away a lot of motor units. And we are calculating the coherence in this very case. Coherence is calculated from the cumulative spike train. And the merged motor units appear to form cumulative spike train in the decomposition results. And we are throwing away cumulative spike trains because they don't come from the individual motor units. And afterwards, we are summing up the spike trains to form the cumulative spike train. Something strange, right? Okay? And here is just an example, a simulation study. Yes, in the fuzzy form muscles, you will very likely see the motor unit merging. And if you take this merging into account, you can increase the number of motor units identified or analyzed. Let's say analyzed, not identified, not to create a confusion, by 300%. Okay? You can still calculate the sensitivity, you can still calculate the precision, but this sensitivity and the precision is now calculated on the level of the motor unit cluster, not on the level of the individual motor unit. Okay. And you can go even further. You can ask, okay, why the decomposition? Because I get rid of the measurement methodology. I get rid of motor unit action potentials. Correct? This is one big step. Compensating for the convolution kernels, motor unit action potentials. This is what the decomposition does. But you can create another quadratic form by playing with the map. You can calculate it slightly differently. So this is a cross correlation between the spike train of a given motor unit and all the channels. But this one is just the vector of EMG measurements at a given time n. So n runs here only with the second post-multiplying vector to define the samples of the spike train. And when you calculate the activity index, n runs on both sides of the quadratic form. Okay? you still compensate the mixing matrix, you still get the quadratic form of the motor unit spike trains, but you calculate it in a different way. And the beauty of this activity index is that now it takes into account all the motor units that were active in the detection volume of the electron. Not just those that were identifiable, not just those that formed the merged clusters, all of them. And we saw in the first part of my talk that the number of motor units matters. And it is the most important parameter according to the analysis that I presented. Of course, I presented it in a way that fits my talk. So don't be mistaken about that. But it makes sense to try to increase the number of motor units. Okay? And we can ask the same question. So I can calculate the coherence between the gastrocnemius medialis and gastrocnemius lateralis. Again, relatively high number of identified motor units. Here is the results of coherence calculation for the different number of the motor units included into the spike train. Please memorize this image, because now the coherence of the activity index comes. And again, in a similar way, in a similar fashion, you can play with the size of the quadratic form. You can extend the activity index in a very same manner as you can extend the motor unit filter by adding the delayed temporal information. Okay? The same mathematical trick applies. And then more you extend, the higher the coherence values you get. Now this image looks quite different than this image. And we need to validate it, of course. We don't fully understand what this means. If I would have 200 motor units, would I see the increase of this coherence to these values? I cannot yet claim this because I cannot validate it. But we can in simulations. But simulations are one thing. Experimental signals are the another. Okay? Okay. Of course, we did some simulations, and you see that activity index performs very well, where it should perform very well, compared to the cumulative spike trains, compared to the amplitude envelopes. The legend is here. For different frequency values, and even in the experimental conditions, you tend to get higher coherence values by using the activity index compared to the cumulative spike train calculated by the decomposition. Okay. This was about the coherence. Kiran gave me 2 hours, so I will use them. We'll finish a bit earlier, okay? Don't worry. But you can calculate the synchrony also by using the temporal measures like correlation, right? And this correlation, when you think about it, is a fundament for many factorization techniques, including the principal component analysis, non-negative matrix factorization. Practically the whole theory of the muscle synergies was built on the temporal measures of synchrony. Okay? So correlation next to the coherence is also very, very, very important measure. In case of correlation, what you do, you can calculate it in different manners, of course. But basically, the story goes like this. So you identify the motor units. Each motor unit is depicted in different colors. I'm sure you know this. But instead of plotting these bars, where each bar represents the firing or discharge of a motor unit, you basically connect them into a line, low-pass filter it, and you get the smoother discharge rates of the motor units. And then you can observe these common fluctuations, and these are the time series that you can use to calculate the correlation. Now you can do this for different muscles, simultaneously active. So this is, again, plantar flexion, gastrocnemius medialis, gastrocnemius lateralis, soleus. These are the expected muscles to be active during the plantar flexion, right? What about the tibialis? Should tibialis be active during the plantar flexion? No, but it was consistently. Why? Because we didn't just ask the subject to do the plantar flexion, we asked them to stabilize the plantar flexion. And how do you stabilize the plantar flexion? By coactivating the antagonists. So, a side take-home message, be careful what you ask the subject to do. Okay? But you see, quite many motor units identified. I deliberately plotted 3 trials so that you can get an estimate of repeatability. I like repeatability because it is an indirect indication of consistency, and consistency matters, okay? And now you can calculate the correlation in 2 different ways. We will calculate it in 2 different ways. First, I will sum all these firing patterns together, basically spike trains. I will calculate the cumulative spike train and low-pass filter rate. Okay? This is in a similar manner how we calculated the coherence. And you have different lines for different muscles, and then you can calculate the correlation between these lines, and you get 4x4 correlation or cross correlation matrix, right? What you can see is that gastrocnemius muscles go together, so this is both gastrocnemius muscle, and that due to the specific task, to this stabilization that was asked for, tibialis and soleus go together, okay? So they are highly correlated, and you observe that, yes, also gastrocnemius muscle have something in common with tibialis and something in common with soleus, okay? So it is not completely 0 here, it is not completely 1 here, but there is a trend. And this trend is repeatable across different trials. And these trials have to be a bit specific trials because they were before the 10 days bed rest. Okay? After the 10 days bed rest, in the same subject, we don't see much change, right? Still the same patterns of the muscle coactivation. But now, let's switch the methodology. This is this old methodology. Now I will cross-correlate the discharge rates of each and every motor unit, okay? So on x-axis now there are motor units, and these red lines depict limits between the different muscles. So this is tibialis, this is gastromedialis, this is gastrolateralis, this is soleus, okay? These are the compartments. And you can get, I will switch now down here, you can see why this is not close, not completely zero. Okay? And the answer is not that you have a behavior which is somehow close to the behavior of the gastromedialis is somehow close to the behavior of the tibialis. It is the part of the motor units behave similarly. Portion of the motor units, some motor units behave. It's not how they behave. It's who behaves similarly to who. Okay? Now, this partial correlation gets a whole new dimension. And after the bed rest, you tend to see a new clusters forming up. What seemed to be more homogeneous activation before bed rest, after 10 days, and 10 days bed rest, for those that do not know, this is on the edge. You would love to prolong it, but the subjects tend to disagree. Okay? So it's on the edge where the things become visible. Okay? And you see that before, we would say, okay, no change at all. But if you calculate it in a different manner, if you go to the clustering of the motor units, then you can plot these force graphs that Daria presented yesterday. Okay? And this gives you a complete new insight. And then you can go and study the repeatability, and more or less you will see that the things are stable. Now the big question is whether these clusters remain stable across dynamic conditions, across electric stimulations. If they are always stable, then this means wiring, and this means anatomy. If these clusters change over time, then this means plasticity. Software versus hardware. I know that you don't like to hear that you are machines, but you are very sophisticated biological machines. We all are. Okay? Mother Nature was an engineer among the others. Okay. Of course, there is a diversity. What I presented in the previous slides was the case of one subject. If you take a look at the different subjects, you will have different results. Therefore, we need to head towards personalized analysis. Okay? Personalized analysis in terms of long-term tracking. What do I have today in the motor system that I didn't have yesterday? And how fast is my motor system getting older? Okay? Long-term tracking. Correlation can also be used for estimation of the coactivation. And we all know that the muscle coactivation is quite strange when it comes to the stroke. Okay? Because the descending pathways are affected. Inhibition is not working properly anymore. We have a pathological coactivation. And here you have a coactivation of the wrist extensors and flexors at the level of the individual motor units that were identified during the flexion, if I'm not mistaken, where only flexors should be active. Maybe it was extension, I don't remember honestly, but it doesn't matter. And again, you can calculate this by the cumulative spike train, you can decompose it, you can sum the spike trains together, and then this is a simulation just to illustrate what is going on. If you do this and if you have a sufficient number of motor units, then you will get estimation of the ramp up in blue line and ramp down, and you can compare it to the simulated force here. If you don't do the composition, then you have a much more variable estimation because the MUAP shapes play with you. Remember, in the second or third slide, I commented on how diverse the contribution of the energy of different motor units is to the surface EMG. So if you don't compensate for this, then you have high variability. But also the decomposition is not without problems. There are decomposition errors that are possible. We commented on that. But also, if you compare the number of motor units as a function of their sizes to the number of identified motor units as a function of their sizes, you can see that the decomposition is biased towards the big high threshold motor units. They are simply the loudest ones. Okay? Even though you start with the exponential distribution, you get more or less uniform distribution of motor units when you perform the decomposition. Okay? Now you can then again use the cumulative spike train, cumulative activity index. There are different ways how you can calculate the activity index. You can read in the published manuscripts, but the end result is that yes, you can get smaller errors by taking into account the activity index. The next one is the cumulative spike trains, while no compensation at all gives you the worst results. Okay? The difference between this blue cumulative activity index and this purple is in the number of identified motor units, as I explained to you. Okay? This is for one muscle. Coactivation means that you compare the excitation of one muscle to the excitation of another muscle. So you divide one by the other. Okay? And if you divide a metric that is highly variable with a metric that is highly variable, what you get is a highly variable metric. Okay? So if you calculate the ratio between the activation of the muscles, then you will get even worse results if you do not compensate for the MUAPs or if you have too few motor units identified. Okay? Okay. And it gets even worse. You can get phantom synchronization. I admit it's a bit strong word, but I have been teaching for 20 years and I know that the strong words are needed. Maybe they are not politically correct, but they are in a good faith of leaving the take-home message. Okay? Phantom synchronization is something which I would call non-existent, methodologically induced. How this happens? Very easily. Again, we have a wrist flexion and extension. During one group of muscles and motor units is active, during the other group of the motor units is active. Okay? This is a healthy subject. No coactivation. Flexion, then extension. Okay? If you apply non-negative matrix factorization, which is frequently or was frequently used to study the muscle synergies, you see one group which is active during the flexion. You see another component which is active during the extension, then you see a third component which is active during both extension and flexions. And this one is non-existent. This one is methodologically induced. Okay? And you can count them. You can count how many motor units are active during flexion and how many during extension, and how many components demonstrate the energy during the flexion and extension. And you can come up with the third group. I will say group 1, group 2, group 3. This one which is in between. So simultaneous activation during both flexions and extensions. And you have one methodology which is the decomposition, and you have so the motor unit identification more accurately, and you have another methodology which is non-negative matrix factorization. What you can observe that regardless of the type of the movement, flexion, extension, radial ulnar deviation, pronation, supination, the non-negative matrix factorization yields significantly more motor components that are active during both phases. If you plot it in another way, you have in yellow the activity during one movement, so flexion, in blue extension, in red activity during both of them. Okay? And cumulative spike train demonstrates ability to separate between those movements, except during the pronation and supination. Okay? This is the cumulative spike. The next column is a cumulative spike train, but weighted with the energy of the MUAPs. Okay? And the next columns are the results of the non-negative matrix factorization, and you can select the number of observed components, 2, 3, 6, 12 in this case. What you can see that the red color, so this unspecific group, unspecific to the movement group, is much more represented when you use the non-negative matrix factorization. And this is due to the fact that non-negative matrix factorization do not compensate for the MUAP shape. Non-negative matrix factorization is not blue. It's not green. It is between the green and the blue in terminology of today's talk. Okay? And you can calculate the same measures. You can calculate the coherence between the cumulative spike train and the non-negative matrix components. And you can ask whether or not they saturate at different frequency bands. You can do this sensitivity analysis. You can ask how representative things are when it comes to the coherence. Okay? And then the things get even worse. In the dynamic conditions, the MUAP shapes change. And these changes are reflected in the components identified by the non-negative matrix factorization. If you simulate the constant excitation and then the shortening and extending of a muscle, if the non-negative matrix factorization would estimate the excitation, then you should see no fluctuation in the estimated components. But you see them. Okay? And Martin, he has a poster, published this, showing that now this is a bit complex graph to understand, but just read these colors. So in this first line, there are the simulations where the excitation range was limited to 20%, from 0 to 20, 20 to 40, 40 to 60. The next one is range of 40, 0 to 40, 20 to 60, 40 to 80. And the next one is range 60, okay? 0 to 60, 20 to 80, 40 to 100. And you see that if the excitation range is small compared to the range of the muscle movement, then you have a lot of muscle geometry reflected in the non-negative matrix factorization components. Okay? And this is methodologically induced. And Matej on Monday discussed that you can get a very, very similar MUAP shape changes even in isometric conditions when the high level of fatigue is present. These are isometric contractions, but long ones. So there is a very clear presence of fatigue. Okay? So the same thing happens. And now we will switch. We will go to the corticomuscular coherence. Okay? And we will start by coherence between the two muscles. It's just because I would like to show you another trick. I'm sure that many of you know it, but it doesn't hurt repeating them. So we have a pathological tremor. It can be Parkinsonian, it can be essential tremor, and we heard a very nice lecture yesterday by Jaire, okay, about the pathological tremor. So here are the motor units identified from extensors, wrist flexors in pathological tremor. You can see this burst of activity. They come with the frequency of the tremor. You can study the basic frequency peak or its first harmonic, etc., etc. But you can also observe the tremor in the different muscles. So these are simultaneous recordings of left wrist extensors, left wrist flexors, right wrist extensors, and right wrist flexors. They were all decomposed. The cumulative spike trains were calculated, low pass filtered, and these are the results. These are the filtered cumulative spike trains. What you can observe is a clear tremor frequency on the left side of the patient, correct? And there seems to be something here which also looks like tremor, but what about this? What about the right wrist extensors? Is this tremor or not? It's hard to tell, but you can always calculate the autocorrelation function. And in the signal theory, autocorrelation is known to enhance the frequency content. Whatever is repeating will be enhanced by the autocorrelation function. And if you calculate the autocorrelation function in this case, what you see is a clear pattern on the left side, clear pattern on the right side. And even though it is not obvious from this cumulative spike train, the tremor is very much present also here. And if you pay attention to the details, you can get the information about the phase of the tremor. Pay attention that here you start with the negative values. This is the edge, right? You start here, but the phase difference is obvious, most obvious on the edge. Here it ends with the negative values, and here it ends with the positive values. This means that they are out of phase. Extensors and flexors are out of phase. And you can apply this phase information also across the limbs, or separately in the left limb, in the right limb. You just need to play a bit of trick on the time series, on the cumulative spike train. And this trick is called autocorrelation function. Okay? I like these old tricks because I would hate them disappear in the era of the deep neural networks. I would hate to see that the era of deep neural networks becomes trial and error era, where the basic principles disappear. Okay? This is a very nice way of saying that I'm old school. Okay. This was essential tremor patient, and this is now a Parkinsonian tremor patient. And again, you could ask, oh, maybe the tremor is also present here, right? If it was present here, it could be present here, but it is not. Parkinsonian tremor in this patient was indeed unilateral. Okay? And there is a very nice paper that we wrote 10 years ago with Juan Gallego, the next speaker, if I'm not mistaken, exploiting this phase information in tremor to say something about the supraspinal centers and the afferent feedbacks in the case of pathological tremor. So I invite you to read the manuscript if you haven't so. Finally, we are at the brain, corticomuscular synchrony, and with this I will finish. I know that this keynote lecture are a bit long. You can calculate the cumulative spike train out of the high density EMG. And then you can use this spike train in a very similar quadratic form to estimate the filter that can be applied to the EEG. So basically, you take this information, calculate the cross-correlation vector, apply this to the EEG, and then you get this component. Now in pathological tremor, so this is the data model. You help yourself with the spike trains from the motor units, but you apply this to the EEG signals. In the case of the pathological tremor, this is very, very successful because the very tremor means that the brains and the muscles are highly synchronized. Okay? And the beauty of this approach is that you get a time series, and then you can apply all the analysis of the time series. You can ask where the tremor is on, where the tremor is off. You can study the dynamics of the tremor. Can you study the dynamics with the coherence? I will leave this answer unanswered. If you do this, you can calculate the coherence. And you will find out that the coherence is significantly enhanced. Here you have the coherence of the component extracted from the EEG. And here, for comparison, you have coherences extracted from the different special filtered EEG channels. Okay? And here is the inertial measurement, so that you see that it's tremor component. And you can study the extension factors. You can now ask yourself, we devoted quite a lot of energy in illuminating the size of the motor unit filters. You can do the same analysis to the size of the EEG filters, okay? And get the answers. And then you can apply this to the young subjects, healthy subjects, the one without pathological synchronization. And you have a poster by Nina Murks illuminating this kind of analysis hanging in the next room. Okay? What you see is that, yes, by applying this EEG filter, the coherence is significantly enhanced, and that the improvements are statistically significant, basically. I will leave Nina to explain this. And then, just to finish my talk, when it comes to the elicited contractions, you would expect them to be highly synchronized. The motor unit behavior highly synchronized, right? Correct? So in this case, we'll talk about the desynchronization. And you will understand what I mean in 1 minute. So when it comes to the elicited contractions, the most common things to study are H reflex, M wave, or compound muscle action potential induced by transcranial magnetic stimulation. Okay? We did them all. We come up with the pipeline that decomposes this. And this is just a short presentation of the motor unit action potentials that form the compound muscle action potential in the H reflex stimulation. If you sum this colored motor unit action potential together, you get this black sum. And then you can compare this sum to the original H reflex, and then you get an idea of how much energy you accounted for by the decomposition. Okay? And you can sort the motor units because this pipeline is a bit specific one. You first perform the voluntary contractions, and then you estimate the filters, and then you transfer these filters to the elicited contraction. Because they are very hard to estimate in the elicited contractions, you do this trick of transfer between the voluntary and the elicited contractions. And the side product is that you know the recruitment threshold in voluntary contractions. Okay? And you can sort the motor units. I apologize. You can sort these motor units with respect to the recruitment threshold in voluntary contractions. And then you can observe the latencies. So this is a stimulus and the latencies of these motor units in the elicited contraction. Okay? And then we would be tempted to say something about the recruitment order based on these latencies. Recruitment order defines the latencies of the motor unit firing, correct? Correct or incorrect? Okay, answer this for yourself. What you see here, that although they are ordered with respect to the voluntary recruitment order, they don't appear ordered in the with respect to the latencies. Okay? Is this a methodological error? Could be. Doubting is always healthy. Be critical. But if you repeat this, now again, I will rely on the consistency, on the repeatability. If you repeat this, if this is an error, then it is a deterministic error. Okay? And you can, this is the results for the M wave. I will show you the results for the H reflex. You can analyze this by superimposing it. So these are the two M waves that are similar in the peak-to-peak amplitude. And these vertical bars are the compositions. So although they are random, they are consistent, correct? So is this, what's going on? Why we don't see the recruitment order in the latencies? The answer is very simple. Conduction velocity is playing with us. Where and when you detect the firing of the motor unit? At the time it passes, the motor unit action potential passes under the electrode. Do you detect it at the very innervation zone? No. Stay away from the innervation zone. And what does it mean that the motor unit, what does it mean, the firing time? When the action potential hits the tendon, when the action potential is generated in the innervation zone, when the motor unit alpha motor neuron gets excited, which conduction velocities should we take into account when talking about the latencies or the firing time? It's a very simple question. But we failed to ask ourselves for 15 years. And it hit us. Right? It was always there. We just never asked ourselves this kind of question. And when it comes to the elicited contractions, when it comes to the study of the synchrony in the elicited contractions, these things matter. Involuntary contractions, they are on the order of a milliseconds, these impacts of the conduction velocity. They are not so important in voluntary contractions. But in elicited contractions, they matter. So you must not, these are the results of the transcranial magnetic stimulation, just to show you that the same problem exists in all three cases, okay? So the recruitment order in elicited contraction should not be estimated from the latencies. At least not for now, at least not in our group, because we don't know how to compensate the conduction velocities. You don't know where the innervation zone is. You don't know how fast the conduction velocity of the alpha motor neuron is. We don't know. What you can do is ask yourself, do the different intensities of the stimulation, and then ask yourself whether the motor unit was activated or not activated at the different intensities of the stimulation. And this is how you define the recruitment order, not by latencies, by activation. Okay? And if you do this in the M wave, then you get expected results, which is a reversed order of activation. But also on this reversed order, I could spend another 2 hours, because it's not demonstrated in each and every subject. Okay? It depends on many, many factors. And they vary across conditions. So to conclude, human motor system is highly complex, involves numerous interconnected neural pathways. Methods used to measure motor unit system have limitations. Some of them are very important, some of them less important. You need to understand methodologies. You need to be able to select the most appropriate methodology. You need to understand its strengths and weaknesses. We will need to understand also deep neural networks much better than we do today. And since the problem is complex, also our knowledge needs to be complex. Again, this is a more politically correct way of saying no pain, no gain. And if you are tempted to learn neuroscience with a generative AI, pay attention. I'm not native English speaker, and I used generative AI to reshape the questions of the quiz that follows. And I will not name it, but you will recognize it, okay? A simple question: which of the following is the primary concern when calculating coherence between the EEG and motor unit cumulative spike trains? Several answers. And then we had a chat. I asked it very politely to reshape it, to make it more clear, to target the student audience, right? I was very happy with the answer. Sure, I'm more confident now in my quiz question because I get an expert help, right? Then, since I'm a teacher, I of course asked, do you know the answer to this question? And I got a very, very, very nice explanation of a completely wrong answer. It's a tool. It's a very useful tool. But we need to have a lot of cautious when using it. Be critical. Doubt it. As you doubt any teacher in any school. With this, I thank you for your attention.